Welcome to this episode of Mentors at Your Benchside, the podcast giving you advice, tips and tools for getting the most out of your research. I'm Laura Grassi, and today I'll be talking to you about the history of cell biology. The cell theory, or cell doctrine, states that all organisms are composed of similar units of organisation called cells. The concept was formally articulated in 1839 by Schleden and Swan and has remained as the foundation of modern biology. The idea predates other great paradigms of biology, including Darwin's theory of evolution in 1859, Mendel's law of inheritance in 1865, and the establishment of comparative biochemistry in 1940. While the invention of the telescope made the cosmos accessible to the human observation, the microscope opened up smaller worlds, showing what living forms were composed of. The cell was first discovered and named by Robert Hooke in 1665. He remarked that it looked strangely similar to cellular, or small rooms which monks inhabited, thus deriving the name. However, what Hooke actually saw was the dead cell walls of plant cells, cork, as it appeared under the microscope. Hooke's description of these cells was published in Micrographia. The cell walls observed by Hooke gave no indication of the nucleus and other organelles found in most living cells. The first man to witness a live cell under a microscope was Anton van Leeuwenhoek, who, in 1674, described the algae Spirogyra. Van Leeuwenhoek probably also saw bacteria. In 1838, Theodor Schwann and Matthias Schleden were enjoying after-dinner coffee and talking about their studies on cells. It had been suggested that when Schwann heard Schleden describe plant cells with nuclei, he was struck by the similarity of these plant cells to cells he observed in animal tissues. The two scientists went immediately to Schwann's lab to look at his slides. Schwann published his book on animal and plant cells the next year, a treatise devoid of acknowledgements of anyone else's contribution, including that of Schleden. He summarized his observations into 3 conclusions about cells. Number 1. The cell is the unit of structure, physiology, and organization in living things. 2. The cell retains a dual existence as a distinct entity and a building block in the construction of organisms. 3. Cells form by free cell formation, similar to the formation of crystals, spontaneous generation. We know today that the first 2 tenets are correct, but the third is clearly wrong. The correct interpretation of cell formulation by division was finally promoted by others and formally enunciated in Rudolf Virchow's powerful dictum, omnis cellula est cellula. All cells only arise from pre-existing cells. Modern cell theory. Number 1. All known living things are made up of cells. Number 2. The cell is structural and functional unit of all living things. Number 3. All cells come from pre-existing cells by division. Spontaneous generation does not occur. Cells contain hereditary information which is passed from cell to cell during cell division. 5. All cells are basically the same in chemical composition. And 6. All energy flow, that is metabolism and biochemistry, of life occurs within cells. As with the rapid growth of molecular biology in the mid-20th century, cell biology research exploded in the 1950s. It became possible to maintain and grow and manipulate cells outside of living organisms. The first continuous cell line to be so cultured was in 1951 by George Otto Gay and co-workers, derived from cervical cancer cells taken from Henrietta Lacks, who died of her cancer in 1951. The cell line, which was eventually referred to as HeLa cells, have been the watershed in studying cell biology in a way that the structure of DNA was a significant breakthrough of molecular biology. In an avalanche of progress in the study of cells, the coming decade included the characterization of minimal media requirements for cells and development of sterile cell culture techniques. It was also aided by the prior advances in electron microscopy and later advances such as the development of transfection methods, the discovery of green fluorescent protein in jellyfish, and discovery of small interfering RNA, among others. The study of the structure and function of cells continues today in a branch of biology known as cytology. Advances in equipment, including cytology microscopes and reagents, have allowed this field to progress, particularly in the clinical setting. Visit the original article for a handy timeline showing the key developments in the history of cell biology. from the development of the compound microscope to the discovery of siRNA. So that's it for our very short history of cell biology. Check out the episode description for links to related articles and resources, and don't forget to subscribe to the podcast to get more help and advice from Mentors at Your Benchside.